welcome Professor Thomas Davison and express our gratitude for his valuable contribution to this seminar on greenhouse gas dynamics in lakes, patterns, and drivers. Thank you, Thomas.
Yes, unmute. Yeah. Thank you very much for the introduction. And thanks for inviting me to give this presentation, have this workshop. I was just thinking about SIL, and actually the first conference I ever went to was an SIL conference in Melbourne. So it's nice that I didn't actually dream I would ever present such a workshop like this when I attended it. And if you'd seen my talk, you also wouldn't have guessed that I would ever present such a workshop. But anyway, here we all are. So I just share my screen. My computer's a little slow.
Oh,
Participants can now see my screen. Okay, you can see my screen apparently.
yes, we can see it.
Okay, great. Then, yeah. So, yeah, as Maria said, I've been working on greenhouse gases. I've been working on lakes and lake ecology for a lot of years now, more than 20, and I've been focusing on greenhouse gases and measuring them and trying to understand them in lakes for the past, 15 years or so, I guess. I started working on greenhouse gases, around 2010, I think 2009, 2010. And I'll talk a little bit now about some general understandings, introduction to the topic, and then at the end, I'll focus a little bit on some of our recent work, some of it published, some of it unpublished. So just a little bit about the background of the situation we're in. So the context being a massive increase in greenhouse gases and methane. And then the left plot, you can see very large rises in the last 200 years. And we have these long-term records from the concentration of methane in ice cores. And in the 80s, we've also seen a rapid increase. And in the 2000s, it seemed like it was flattening out a bit, but it's since increased again. Although the rate of increase is slowing a little bit. And then we have CO2 measured in the center of the Pacific Ocean in Mauna Loa Observatory in Hawaii. And there we see the steady increase in CO2 concentration, which doesn't actually show any sign of slowing down. So this is the context of the way I think about greenhouse gases in lakes, and can we minimize their emissions, and can we maximize their influxes? Is that possible? Is that something we can even think about doing? And what I will talk about here today mostly is exclusively is the three main greenhouse gases that are emitted from lakes. So this is carbon dioxide, methane, and nitrous oxide. And I talk a little bit less about nitrous oxide, largely because it's much more understudied than the other two. And these are some of the different types of lakes we work on. I think one of the cool things about ponds in particular is that they are easy to manage. For example, this pond here in the middle is actually the same pond as the one in the bottom after a relatively straightforward restoration exercise. So yeah, that's kind of cool to see that you can make such a large difference with such a small input. And just to say that there are many types of lakes we work on and that can be studied. On the right-hand side here, we have a 1 million-year-old impact crater lake in Ghana that we have worked on a little bit, Lake Pasumtri. And here we have a quite typical Scandinavian lake. And here we have permafrost systems and the Greenlandic lakes. And again, my favorite restored pond at the top. One last one, I don't know why that was animated, but that's another favorite lake of mine. That's a lake from Northwest Greenland, where there's a large colony of birds in the catchment, which affect the chemistry and biogeochemistry and biology of the system. So, why study lakes? Why study greenhouse gases and lakes? The lakes are a key part of the carbon cycle, despite their small percentage of the land area. And I did have a number in here. I watched a YouTube video from an American publication yesterday, which said it was 1% of the land area, but it really depends where you define a lake. If you go down to very small water bodies, then you can come up to higher than 1%, more towards 3%, I think, of land area that could be defined as covered by lakes. and they have they're important in carbon dynamics, and as I said just now, they have the potential to be a sink or a source, where you have inputs of groundwater into the lake, and you have various processes within the lake, which can lead to the production of CO2 and CH4, so carbon dioxide and methane, through the mineralization of organic matter. You also have carbon burial, you have export, you have photomineralization of organic carbon, and then you have emissions of carbon and methane from the lake surface. And in a really classic paper led by Tranvik, published in 2009, they proposed this idea that lakes and reservoirs were regulators of the carbon cycle and climate. And they introduced this concept of the freshwater pipe. So you have these 6 pipes here for different lake types. So you have inputs of dissolved inorganic carbon, dissolved organic carbon, and particulate organic carbon. And then it's processed by the lake. Some of it is stored, some of it is emitted, and some of it is exported downstream. So putting numbers on these different parts of the pipe is incredibly important if we want to understand how lakes process greenhouse gases and particularly carbon within the landscape. And you can see they have very different numbers coming in here. So in different types of lake, in a hard water lake, they say they have a CO2 influx, a lot of carbon storage, and relatively large output downstream. And a floodplain lake would have a very different behavior in Brazil, would have much more higher input of organic carbon, might be particulate organic carbon. So where the lake lies in the landscape will depend what its inputs will be, and what are the processes and factors and parameters that control this processing is one of the things I think I'm most interested in studying, and have been studying the last few years. But one thing missing from this paper, and most freshwater studies for the last years, but unfortunately it's changing a bit now, is that there are no ponds in this study, no small water bodies. And they can be incredibly important for the processing of carbon. And they are quite different from all these systems in that they're normally closed systems. So they don't have an export in terms of surface water, at least, although they lose water to the atmosphere, of course, and to maybe to groundwater. Okay, so we need to look, just want to introduce a little bit what the different parts of the sea cycle and the sinks and sources for the different gases we're going to talk about today. So this plot has both methane and carbon dioxide in it, and there is this classic picture of a lake, a stratified lake. So we're thinking about a deeper lake here with a hypolimnium and a thermocline and epilimnium. And so the hypolimnium and the epilimnium are thermally discrete, so they're separated. So you need to have diffusion across this thermal barrier. So the waters do not freely mix. So it's a process of diffusion rather than free mixing, which controls the exchange. And you have this various different processes that lead to your concentrations of dissolved CO2 or partial CO2 or partial pressure CO2. There's lots of different, one of the things about greenhouse gas studies is there's a lot of different ways to say the same thing. But here we have the organic matter in the epilimnium being processed to mineralized to dissolved CO2. And depending on pH, alkalinity, then you get this converted to carbonate, bicarbonate, and then precipitated perhaps as calcium carbonate, where it can be sedimented. Again, it can be dissolved. So there's the number on the processes. We have carbonate precipitation here, and we have photosynthesis. No, not photosynthesis. We have we have photosynthesis taking up the dissolved CO2 into the organic matter. Then the organic matter settles out and can be sedimented and be converted to dissolved CO2, increasing the parcel pressure in the bottom waters. In this traditional model for methane production, we have methane being exclusively produced in the sediment when you get anoxic conditions. You also have SO2 reduction, which can lead to carbonate. So you have these different chains of processes going on in the epilimnium, the hypolimnium, and you have the surface inlet and the groundwater providing DIC and DOC for these processes. So it's a web of factors going together, being controlled by the processes within the water column that all together lead to the emission or consumption of CO2 at the surface, and the burial of carbon and sedimentation of carbon in the bottom of the lake. So then if we move on to methane, exclusively. Methane is a bit different from CO2. CO2 is very soluble in water, so we really only get dissolved CO2 in the water. So when CO2 is produced, it's dissolved. Yeah. With methane, we have quite a different situation, as you do actually with oxygen as well. Um, when both methane and oxygen can be rather insoluble in water, so they can form bubbles. When the production rate of methane is higher than the dissolution rate, you get bubbles formed, and these can be periodically released from the sediments. In the deep waters as well, once it gets too deep, the pressure rises and bubbles don't form because the dissolution rate is rather high at pressure. And in the shallow parts of the lake, you also get bubbles released. And this is a process called ebullition. You also get methane. If it's produced slowly enough, it diffuses into the water and becomes dissolved CO2, CH4, which is then can be trapped in the bottom waters and pass across the thermocline by diffusion and can be consumed in the water, but I'll talk a little bit about that in a minute. And this is so we need to sample methane in different ways to carbon dioxide. For carbon dioxide, you can sample a flux with a chamber. You can sample all the gases with a flux in the chamber, taking samples over a period of time and calculating the change over time so that you get a flux rate either in or out. Can be both for CO2. Can even be both for methane sometimes, and also now I think about it, can also be both for nitrous oxide. But in terms of sampling the dissolved concentration and the diffusion emissions and sampling the bubble emissions, you have to take different approaches because bubbles happen sporadically, very unpredictably, and so you need to be there all the time to sample them or for a longer period of time. So these bubble traps have been used in the past, classically by Wick et al. In 2016. They published a very nice paper on bubble emissions from Arctic ponds in Sweden. And then if we look at some results from sampling methane in the water column, this is from 3 different lakes of different depths. This is the depth here on the left. And then we have concentrations of oxygen. Now this is temperature in the squares, oxygen in the circles, and then methane in the bottom waters. So you can see Pete, this is the classic North American lakes, a paper led by David Bastviken with Cole and Pace and Bogart, and they sampled the methane concentrations at different depths in 3 different lakes. And here we can see that we got very high methane concentrations in the hypolimnium, and once we get oxygen in the system, the methane concentrations start to go down and are very low in the surface waters. They also measured methane oxidation rates in the water column, and here we can see that the oxidation rates within the water column were much higher once you get up to the oxic zone. Methane oxidizing bacteria, it's the clues in the name, although you can have anoxic methane oxidation, require oxygen and methane to do their thing. So once you get this zone where you have both methane and oxygen, the methane oxidation rates can be very high to actually reduce the methane values to be very low. So here we have the seasonal measurements of the methane in these different lakes, and we can see there's quite a bit of seasonal variation. And if you see the close-up into the surface waters, you can see that there's an increase in methane as you get towards the surface waters. And what the authors suggest here is that this is actually methane being mixed from production in the littoral areas, that in the deep water zones, all the methane is consumed before it reaches the epilimnium. But in the epilimnium, the methane produced in the littoral areas of the lake is mixed into the entire water column and mixed together. So yeah, to summarize what they found is that 51 to 80% of the methane produced in deep sediments was oxidized in the water column. Most of the sediment released from shallow waters escaped oxidation and reached the atmosphere, and that's what circulated around into the surface waters. So actually, you can have quite high methane production rates. These values in micromoles are rather high, but it was all consumed by oxidizing bacteria before it reached the sediment surface. Recently, there's been an idea of oxic methane production, and it comes from work in Germany on Lake Stechlin, a lot of it. And here they found that they had this peak in methane concentration in the thermocline. And it's a little bit controversial. There've been comments on this paper saying is actually the methane is literally produced and the currents you can find papers in the literature for both arguments, but it's indisputable that we get this peak in methane concentration here at the thermocline with a peak in oxygen and a peak in algal productivity. So there's this increasingly prevalent idea of methane being produced in oxic waters in association with high productivity for this subsurface chlorophyll peak in these rather oligotrophic or less nutrient-rich, stably thermally stratifying lakes. And then recently in 2022, there's this paper on methane, oxic methane production in small temperate lakes, but there's not been that much of this studied. Um, and then they say that there are different drivers of the oxic methane production rate. But yeah, you can see that these 4 points at the top, which are a different lake, are doing a lot of the work in these regressions. So I'm not totally sure about this, but there's some studies showing that you can have oxic methane production in small temperate lakes, and that as the surface mixed layer volume, so as the volume of water increases relative to the area of the sediment, you get an increasing amount of methane, oxic methane contribution in these small water ponds. And then finally, we have nitrous oxide production in lakes or nitrous oxide consumption. And this is something that has been increasingly worked on a little bit more now. And this paper is basically suggesting that we will have increased N2O emissions for inland waters. It's quite hard to figure out what actually drives emissions of nitrous oxide from inland waters. It seems to be mostly the availability of nitrate from what I can see in the literature. And there was this quite cool study in Canada published in PNAS, which found that ponds and small water bodies are actually unexpected sinks for nitrous oxide, and that it's a bit associated with increased chlorophyll production, that you get increased nitrous oxide consumption in ponds. And again, they look at the effect of oxygen saturation on the different processes which lead to nitrous oxide. And I don't have time to go into what the nitrous cycle is, rather complicated, and we don't have time to go into the details here, but it's a great paper to look at if you want to get some further understanding of it. So what are the patterns that we can observe in emissions that cause the differences between lakes? And one of the things we're of course interested is the effect of temperature on emissions. And this is a nice study from the 2000s by Sebastian Sobek and others from Uppsala, where they looked at partial CO2 pressure on a very huge range of lakes, collected data from many different studies, and looked at the concentration of PCO2 in the water to see if there was any temperature effect. And the title of the paper rather gives it away, as this temperature independence of the carbon dioxide cycle in global lakes. The red dots are the repeated observation in a given lake, and then the blue triangles are the mean for that lake. So you can see if you take the seasonal data, you do see a slight increase in PCO2. There's a huge range of temperature, annual mean air temperature. That's really a lot from Arctic to tropical, and you can see there's something there, but it's not a strong relationship. There's very little predictive power. What they did find had a better predictive power, but again, it's not the strongest relationship. But when you look at the means, it is. But there's clearly something else going on. Is that the log of dissolved organic carbon seemed to be a relatively good predictor of PCO2 concentration. And then we have to think about whether size matters for lakes. And the short answer is that it does. In a nice paper from 2016, Holgerson and Raymond really nicely demonstrated with these plots that the area of the lake, this is the ratio of CO2 to methane, this is methane concentration, and this is CO2 concentration. So the effect on CO2 concentration wasn't so marked, but this is a log-log relationship. So over here on the left, the smaller lakes tend to emit more CO2. A much stronger relationship was methane at the smaller lakes. The slope of the regression is much larger, so the effect of size seems to be much greater on methane emission from so small lakes are emitting much more methane, and also their ratio of CO2 to CH4. So the amount of CH4 produced for a given amount of CO2 is also much larger in these smaller lakes. And yeah, they estimated that lakes under 0.1 hectare, where 9% of the standing water, but 15% of CO2 emissions and 40% of diffusive emissions of methane. So this is just diffusive emissions as well, not including ebullition. So is that an important finding? And if you look at the number of lakes for a given size, so this is size of lake. On the left, you can see that the number of water bodies of these small lakes is huge. It's by far the most numerous pond lake type on the planet. And also they account for these bottom three sizes under a hectare account for I know that's a kilometer squared. These two smaller sizes are dominant in area as well for globally. So what's happening in these small ponds and lakes is very important. And then so size matters, but does trophic state matter? So this change if we talk about shallow lakes at least, this clear water condition with plants to a more turbid state, or this increase in chlorophyll, if you like. And then a study by Del Centro and others um, uh, proved that trophic status is actually very important for determining CO2 flux with an increase in CO2 flux with TP, but that it was dependent upon lake size. So with large lakes, you could actually get a decrease in CO2 efflux with TP. So there's several things going on here. So it's not that straightforward. And then in contrast to Holgerson and Raymond, they found that surface area didn't seem to have an effect on methane emissions. So it's obviously not that straightforward. Depends on your data set a little bit, but they found a strong increase in methane flux with TP. And this is one of the few studies to include nitrous oxide, as they found an increase in nitrous oxide flux with chlorophyll A and an increase with surface area of lake. So that's a little bit in contrast to the findings of the PNAS paper I just mentioned. So what controls this emissions? What controls the processes going on in the freshwater pipe? And can we use some of the theories of ecology to predict these things? So this is a study from Yvonne de Rocha, and I think this one was in the Proceedings of the Royal Society, looking at the different activity energies of the different processes that go on in lakes, activation energies, I mean. And activation energy is an expression of the increase in metabolic rate with increased temperature. So on the bottom of this plot here, we have temperature. It's actually an inverse of temperature, so it's a higher temperature on the left and a lower temperature on the right. And you look at the slope, and these are the different slopes of net primary production, gross primary production, and ecosystem respiration. So NPP is this one minus this one. So it's an expression of how much increasing temperature will increase respiration rates and increase primary production rates. And the difference in the slope means that respiration will increase more with increasing temperature than gross primary production. And what that means is that CO2 emissions should increase with increasing temperatures as respiration comes to dominate gross primary reduction. And then if you look at this is the same thing applied to methane emissions. So the data are quite played around with here. They're standardized, and methane emissions are standardized for a huge array of ecosystems. But the take-home message is, and unfortunately they reversed it here. So this is lower temperature, this is higher temperature. So what this means is that according to the theory of a metabolic theory, as you increase temperature, you increase the rates of respiration more than the rate you increase the rates of primary production, and you actually increase the rates of methane production more than the rates of the other two. So with the theory, with warming, you should increase methane production, increase respiration, increase methane production the most, increase respiration the second most, and increase primary production the third most. So you shift the system over towards methane production and respiration and increase. This would result in an increase of greenhouse gas emissions. And this is for aquatic ecosystems, wetlands, and rice paddies. But you also have other things that are affecting methane production. And this is the paper from another paper where we show that chlorophyll A production increases methane emissions, both diffusive and ebullition. This is one of the first studies that tried to include both diffusion and ebullition into the calculations. So you have to be a little bit careful, I think, applying this metabolic theory of ecology, that it's a little bit of a simplification. And one of the reasons is, we'll just jump over this bit, is that the data here are not just an average of the site. They are data from many sites from winter to summer. Is it actually a seasonal temperature gradient along this axis? It's not a difference in annual average temperature. If you look into the supplementary material of this paper, you can get to the average annual site temperatures. And here you can see that this is aquatic systems, natural wetlands, and rice paddies. This is mean emissions of each site, and this is a standardized emission, standardized by temperature. So an emission estimated to take place at the average temperature of that site. And you can see when you take average site temperatures instead of seasonal temperatures, the relationships in some times still exist and are still significant. That temperature still predicts mean emissions in aquatic systems and in natural wetlands, but the relationship is not very strong. It has an R squared of 12. It's explaining only 12% of the data. So with seasonal data, you can make it look like there's a much stronger relationship between temperature and emission. But if you start using average site temperatures and mean emissions or temperature standardized emissions, you can see that there must be other things controlling those emissions. So I was interested to compare this like seasonal temperature gradient with an actual temperature change. And the place that we can do that is experimentally in our ecosystem, in our mesocosm setup that we have at the University of Aarhus University. It's near a place called Lemming. It's not the animal, it's a place. And here we have a mesocosm setup with 24 small artificial lakes. The system's been running since 2003, so it's very mature. The relationship between the warming treatment and the sediment is in equilibrium. We have two different nutrient treatments. So we have high nutrients where there's a plus here and low nutrients where there's no plus. So the B, C, E, and H are low nutrients. And then within each nutrient treatment, we have three temperatures. So we have an ambient temperature. We have A2 is the treatment in green, which is warm 2 to 3 degrees across the season, depending on the differences larger in spring and autumn. And then we have a 4 to 5 degree rise. So here we have a seasonal temperature difference and we have an actual temperature treatment difference. So we can kind of see how they compare. And this is just to show you the difference between the low and the high nutrient tanks. In the low nutrient tanks, we have clear water and plants, sometimes some filamentous algae. And in the high nutrient tanks, we have a turbid system. They're not always this turbid. This is just some good blue-green pictures which happen occasionally. But there is the general difference between low nutrient and high nutrient that there's yeah, the one is turbid and algal dominated, and the other is more likely to be clear with plants. And that's just it was a reminder of the seasonal difference, and this is actually what we found in our study in 2015 that controlled the diffusive emission of nutrients. It was not related to temperature at all, temperature treatment, but actually in the low nutrient tanks, it was related to plant abundance, CO2 emissions. This is, and in high nutrient tanks, it was related to chlorophyll A. That's kind of what you would expect. And that the methane emissions could be best predicted by ecosystem respiration, but also plant abundance. So on the left, you have low nutrient tanks, and you can see that as the ecosystem respiration goes up, you have higher methane emissions. But these symbols with the larger the symbol, the higher the plant abundance. So the model in the end included both both ecosystem respiration and plants had a negative effect on methane emissions. And there were sometimes plants in the high nutrient tanks, and they actually had a slight positive effect on the dissolved concentrations, but that's a different story. Come back to that. Just to note that if anyone is interested in the effects of plants on regulating the methane cycle, there was a great special issue in Aquatic Botany earlier last year, actually now 2022, on the role of plants in regulating aquatic methane fluxes. So we also studied the effects of that study was looking only at diffusive emissions, and then we realized we couldn't really do that without looking. It wasn't a good thing. We needed to check out the bubbles too. So we put these bubble traps in the tanks for a year, and we measured bubble emissions and diffusive emissions. And we also made a separate study and to come back to the metabolic theory of ecology idea. We could also make this seasonal gradient and create an activation energy from our data. If we use the seasonal data, looking at methane emission, both ebullition and diffusion combined, there was a difference between the high nutrient in green and the low nutrient in blue. So high nutrient tanks had a larger emission in keeping with the plot you saw earlier about chlorophyll A being to predict better a predictor of methane emissions. So we could create this activation energy based on our seasonal data, but we had there was no significant temperature effect on emissions, not alone at least. What we actually found is that there was a massive increase in bubble emissions when you combined warming um, and and nutrients. So these are the total annual bubble emissions from the tanks. But combined emissions, actually, this is diffusive emissions at the bottom and bubble emissions in the orange. And you can see there's no temperature effect in the low nutrient tanks. You have less emissions actually at the intermediate temperature, and you also have fewer emissions here, lower emissions in the highest temperature tank in the high nutrient treatment. But there was a synergy between warming and nutrient addition, which caused a huge increase in the bubble emissions. And actually, up to 99% of methane emissions were by bubbles in the high nutrient, high temperature tanks. So there's a take-home message that it's very important to keep in a warming world, it's maybe very important to keep our nutrient levels as low as possible and try and retain the ecological quality of ponds and lakes if we don't want to emit large amounts of methane. So we showed that experimentally, and then a very nice paper came out also by Aben et al. In Nature Communications, which also showed across real sites, not just experiments, that there was an effect of temperature on ebullition. This is on separate mesocosm experiments. They had a mesocosm experiment there too, but they also had some ponds and some lakes. There is a little bit of the seasonal effect going on here too. In summer, you have higher bubble emissions, but they also nicely showed it experimentally that they had an increase in bubbles with warming, although there's no real eutrophication gradient in this study. So we did a separate study again, and we did a short-term study then to look at the control of bubble flux on mesocosm experiment, and we looked at the types of dissolved organic matter. And what we found was that all these things, DOC correlates very nicely with bubble flux. Plant abundance had a very negative correlation with bubble flux. Here we have macrophyte abundance in the top left, and DOC had a very positive relationship. But the microbial substances most associated with algae and that are produced by by degradation, by the rotting basically of algae as they sink to the bottom of the lake. And this is pond and the mesocosms. This was measured in the water column, not in the sediment, but still, so the supply of fresh labile DOM seems to be the thing that most controls the bubble emissions on a short-term scale, at least in our mesocosm experiments. So, kind of to try and summarize the real-world factors that shape greenhouse gas emissions and their possible feedbacks with climate, there is this really nice figure from Marianne Meerhof's paper published last year, where we look at warming and how it will change various processes in the catchment, including fire and a greater input of DOC from the catchment, maybe more storm events, increasing nutrient input, more organic carbon, and how this will affect the various processes in the lake and how this might or might not. So the effect of, for example, greater cyanic bacterial blooms on net CO2 balance is unclear, as they will increase CO2 intake during periods of high productivity. But what happens when the blooms decay? And there's even a little bit of evidence that blue-green algae can directly produce methane whilst they're producing, and what might stronger stratification patterns do to lakes? Yes, patterns and processes controlling greenhouse gas emissions from lakes. And then I'll give you finally, I think have a little time, 10 minutes or so left. I'll give you an example of some of the studies we've done recently looking at some of these things. So the background to this study is that climate change will increase heat waves, um, and this will increase periods of lake stratification in the future. So it's a very nice paper by Allway in Nature and from 2021 showing what future climate change will do to lakes on a global scale. So the increased duration of heat waves will increase the duration of lake stratification. And there's kind of two possible ways lakes could go when they're stratified. And of course, it may depend to some extent on morphometry and enrichment. But this study, Hot Tops and Cold Bottoms, set out the alternatives, and it suggested that it could be prolonged periods of anoxia in the bottom waters could decrease CH4, decrease sea burial, increase methane emission, whereas prolonged periods at lower temperatures could increase sea burial and decrease CH4 um, emissions. So this kind of shielding, cooling of bottom waters might reduce emissions, whereas this prolonged anoxia might increase emissions. And this modeling paper suggests synergy of warming and increased DOC and nutrients will reduce the epilinium thickness, reduce bottom temperature, and so shielding will dominate. But there was no ebullition in this model, and also yeah, and it's a modeling exercise. So it was interesting, and it's good to pose questions through modeling. But to my mind, there's no real substitute for empirical data, and that's what I like to do. And so we have the possibility to work on this lake near Sukkerborg in central Jutland in Denmark, and it's a relatively shallow. Maximum depth 5.5 meters, mean depth 2.3, long hydraulic retention time, extremely high phosphorus, but not relatively low chlorophyll for the phosphorus level, but it's a turbid lake with very few plants present. And we have a detailed depth map, so you can see large areas of shallow around 3 meters and down to 0.5, and then one deep area where we were lucky enough to put a high frequency buoy measuring temperature every 15 minutes, so we can track the stratification patterns. And this is the stratification patterns from 2020, where we had this is the lake depth and this is the lake temperature. So in June, we had a extended period of stratification. Then we had mixing, short stratification, mixing, and then a longer period of stratification in August. And this is the dissolved oxygen measured at 3.8 meters. So it was anoxic for June, and then when it mixed, you get oxygen back to the bottom waters. And as it stratifies and mixes, you see the oxygen becomes depleted in the bottom waters. So over this time, we measured bubbles using a series of floating chambers, which have some drawbacks. There's better ways to measure it, but they provide a reasonable estimation of bubble emissions. And what we can see here on the bottom is that these gray periods correspond to periods of stratification and the white periods to periods of mixing. And the reason the lines are flat is because the sample is collected every 2 weeks or every 1 week, and so we estimate the bubble emission as milligrams of carbon per meter squared per day for that period of sampling that preceded the collection. And you can see where the different transects are. So transect 1 is at the deep point, transect 2 is in the shallowest area, and transect 4 is 2, and transect 3 also goes into a relatively deep point. And so you can see that in the periods of stratification, we have much higher levels of ebullition than we have in the mixed periods. You actually see this more detailed here. So when the samples are green, it means the lake was mixed. So this is a sample taken on the 27th of May. So here we have a range of 0.1 to 26 milligrams of CH4 as carbon per meter squared per day. And then on the 30th of June, which is the end of this period, we had a peak in emissions ranging from 2.3 to 189 milligrams per meter squared per day. And then a week later, on the 14th of June, this point, after we'd had 2 weeks of stratification, we're back to very low bubble emissions. But it's notable that the emissions are highest actually at the deepest points. So these points where the effects of stratification will be first felt seem to have the largest ebullition. And then if we look at the dissolved concentrations, we can see this is the surface waters, and this is micrograms per liter of methane. And we have three sampling points, the deepest point, the middle of the lake, and the shallow area. We can see that we had quite similar levels, apart from just the day before this mixing event here, the day before the lake started to mix here and before the shallow area. So perhaps we get some upwelling of methane-rich bottom waters here in the shallow point, or we get high methane production in the shallows here. But what's notable here is that in this period of stratification, we got enormous amounts of methane building up in the bottom waters. So this is milligrams per liter, whereas in the surface it's micrograms per liter. So this is 5,000 of this unit. Yeah, and then when we come back a week later and sample again, it's down to 0. So the mixing event uh, happens over actually a couple of days, it looks like, in this site. So you would need to come and sample every day to capture the mixing event correctly. But we tried to estimate the amount of methane emitted at this point of turnover by using the concentration in the bottom water and the depth of the hypolimnium to calculate the weight of methane that was actually in the hypolimnium at that point. CO2, you see a similar pattern. It's a bit more variable in the surface waters. The surface emissions do not seem to be structured by or at least we can't see it from the data resolution we have. They're not structured by the patterns of stratification in the lake. Whereas in the bottom, they are. You get massive buildup of CO2 in the bottom waters as the lake is stratified. These are the same units, though. It's notable. So in the bottom waters, the ratio of CO2 to CH4 changes dramatically, and so we're overall without oxygen. There's not so much CO2 being produced. What CO2 is there is probably a result of anaerobic methane oxidation. So this is kind of the summary of the processes going on. But the differences between a stratified and a mixed system, that you have this big buildup of methane and it becomes a similar volume actually as the CO2. And when the lake turns over, this is released, and that we have many more bubbles, much higher rate of bubble emission of CH4 in the stratified period. So from our results, I mean, this paper's actually will not be discussions anymore. It's just been accepted and will come out in Biogeosciences in a few weeks, hopefully. So we see that the mixed period has a much lower greenhouse gas emission in all particularly in bubble emissions, but that the turnover flux is something it's very difficult to measure, and we have to think about how we would do that. So this is really a summary of that in terms of the total emission of the gases by diffusion, ebullition, and turnover, and this is in CO2 equivalents. So that we can see that turnover flux of methane in this system estimated the way we estimated, which contains some inaccuracies for sure. It may be a bit of an overestimate because we take the concentration in the bottom water and we don't assume it's all emitted. We assume that some of it is oxidized, but it would be better to have surface measurements over these days. But anyway, the take home is that if you went to this lake and just put a chamber on it for a few days every month, you would only measure this and some of this, and that you would miss these other parts of emissions. So you would accurately characterize CO2 emissions, but you would make a very bad job of measuring methane emissions. And this is just to note that we've actually been measuring at this lake for 4 years now, or 3 years now. And at the end of 2020, there was a biomanipulation to try and improve the water quality of the site. So the zooplanktivorous fish were removed, and it had effects for a couple of years in that it reduced the methane emissions, it reduced the chlorophyll. But this last year, I'm afraid, it's gone back to as it was, or actually even worse. Which you can see again from this plot, which is the total ebullitive emissions from the growing season from each of those years. So it's actually worse this last year than it was in 2020. So I think I will come to close to the end, and this is just to present some data from a recent paper, and this is, I think, one of the challenges we have in lakes and freshwaters, is that it's really hard to measure greenhouse gas emissions accurately. This is a kind of a state-of-the-art paper from Nature Geoscience looking at methane emissions from all sorts of ecosystems, not just, it is aquatic ecosystems actually, and it showed that half the global emissions come from highly variable systems. And the highly variable systems mean that the mean and the median are very different. So in wetlands here, the mean and the median are actually very close. But if we look at streams, the mean and the median are quite different. And in particular, if we look at lakes and ponds, the mean and the median are very far apart. The median is much lower and the mean is much higher, which means that we have some sites which emit really loads more than others, and that it's the distribution of emissions is very skewed. And this could be because similar sites emit very different amounts of methane. But I also think it could be because we measure so badly that we all do. It's not a criticism of anyone in particular, but if you go once a month and measure, which is what these data are based on, you can see from the measurements at Ormstrup that you would get very, very different measurements of bubble emission if you came if you came once a month. You could get all the peaks or you could get all the troughs. And depending on where it was situated, you could estimate emission to be more than 50% different. You could have orders of magnitude different. So this is just uh, to state that we need more measurements and we need better measurements. And one of the ways to do this is to try and make high frequency measurements. And there's a possibility to do this thanks to work by David Bastviken and his group. They've developed the application of cheap, low-cost sensors, which cost in the region of hundreds of euros to make a system that can measure greenhouse gases, and they've proved that they are accurate down to atmospheric levels. And we have been lucky enough. Then you have to put them in the chamber, and then you have to think about how to flush them because you can't just leave them on the lake not flushing all the time. You need to flush them with fresh air to get flux measurements for CO2 and methane to separate diffusive from ebullitive flux. And we have been lucky enough to develop a system here that we've applied in this last summer. So we have a cheap CO2 sensor and a cheap methane sensor in here, and electronics is in this box, and it's run by solar, and it measures on a 4-hour cycle. So this is the measurement of methane of a 4-hour cycle from 9 till 1 in the morning. On which date was it? On the 23rd of August. And you can see this is this is diffusive flux, and these are the bubbles arriving in the chamber. And then this is it being flushed after 4 hours. So this is an example of the kind of plots you get. This is from our mesocosm experiment. So here we have the CO2 every 4 hours being flushed back to atmospheric levels. So we have an influx of CO2 and an outflux of methane. Methane is the orange one. And here are the bubbles arriving. So we developed a system to separate the ebullition from the diffusion. So this is this black line that just appeared here is the total emission over 4 hours. And then we can take this line as the diffusive flux and we extend it to the 4-hour period. So we have ebullitive flux is this amount and diffusive flux is the amount where the dot goes to. Of course, if you have a lot of bubbles arriving, it's much harder to do it. So we've developed a system where we randomly sample 100 times over short periods. And then we take the median beta, which provides us with a good estimation of diffusive flux. So we can do it even in times when bubbles are arriving very frequently. And then I'll just show you very quickly that we applied this to a lake, the same lake in Ormstrup, and we get these daily variations in ebullition in the blue and diffusion in the red. So this at the end is a period where the lake was stratified with very little, quite a lot of ebullition and not very much diffusion, and then it turned over a bit here, so the diffusion became much higher. And this is another period where we applied it and we got a huge ebullition event. Normal amounts of emission are around 50 to 100. We got a 1,500 level of emission when the lake really turned over and we had got a lot of ebullition. And we can show why this happened because the black line is the atmospheric pressure. So you can see when atmospheric pressure drops, we get ebullition events. In particular, this very low drop here in September caused a massive ebullition event. So I think I'll skip over this one. Because I think I've used up my time. But just to summarize that, yeah, there's still a lot to do to understand and measure greenhouse gas emissions from lakes and ponds and freshwater systems. Generally, we know a lot already, but there's still a bit to do. So thank you for your time. and your attention.
Thanks to you, Thomas. Very interesting and excellent presentation. Thank you so much. So we invite everyone to write your question on the chat so we can read them and Thomas can answer it. So I don't know if I'm not seeing the questions. I don't know if someone is writing. Okay. So in the meanwhile, because they are shy, yes, they don't dare to ask any
More
question.
confused.
They are thinking. No, I mean, this type of studies represent a great effort in terms of maybe technology to make the correct measurements, because as you say, if you don't go to the lake continuously, the patterns you find could be very different. And in a way, when you show that slide, what the difference between the mean and the median is showing somehow that, and also that different lake with different traffic conditions and size can behave very differently, right? So for you, what would be the future lines of research, considering the heat waves that we are facing and the traffic states of most lakes, especially in agricultural areas?
Yeah, I mean, it's hard to measure, to be there and measure all the time. I think we have to try and use technology to do that. There's many studies have gone and measured once a month or even less frequently, and they have produced data of some value. I mean, there's been a lot of good work done. I don't want to talk it down. It's great. There've been many great studies, but I think what we can see, particularly from shallow systems and systems that stratify and mix, like are not stably, if a system stably stratifies over the summer, I think it's not so important to go so frequently. But if you have a system that mixes, then you can really totally underestimate emissions. So I think it's very important to know your system and to design your study well. But of course, it's not possible to measure everywhere all the time. So we need to do something. We need to be able to. We need to develop ways of modeling it so that we can upscale results, so we know what the controls are, and we can upscale them so we can make predictions about what would be the best management of a lake or pond, or how we can best construct them or restore them. And in terms of nutrient enrichment or trophic status, I think in terms of everything, in terms of biodiversity, it's just it maybe previously there was things published in the literature which said that, eutrophication was actually quite good for carbon burial, for example. It increases carbon sequestration and storage. But what we can say is that that's a bit of a false. I don't think that's a good idea to enrich every lake and pond so that we increase carbon burial, because we just increase methane emission. And maybe we make CO2 emission more variable. So I don't think this changes what guidelines for management of fresh waters. We should try and reduce nutrient enrichment as much as possible and protect our fresh waters from agricultural land use as much as possible, by buffer zones or by separation, or whatever is the most appropriate management strategy.
We have some questions. Shim asks, we will also look at stratification and methane fluxes and found similar results. We have also found that floating plants can have a huge effect on dissolved oxygen levels. Have you looked at the effect of floating plants like duckweed?
This is a really, really great question, and the short answer is no, but I would really like to. But I've read yeah, we have studied some ponds with duckweed, but not in a specific context of looking at duckweed. But it's really hard to study with traditional methods because when you put your chamber on the duckweed, it causes all the bubbles that are trapped under the duckweed to be released, and it's really, really hard to measure in these systems. I've even seen a cool picture of a methane bubble on the surface of the water covered by duckweed and trapped under the surface. So I think you're totally right. We need to find measures of measuring under there because this would cause huge anoxia, and it must be underneath the duckweed layer. And I'm very curious as to what that does to the biogeochemistry.
Ivor asked, thank you for your presentation. What do you think about river reservoirs for hydropower plants? There is a chance of strong evolution.
Yes, there is. Yeah, and there've been several studies from reservoirs showing strong evolution. It would be interesting to see if there were any systems of management of the water level to control that. It's also, I mean, I think it's really hard because the highest demand for water is obviously in the summer, and then you reduce the water level, you'll reduce the pressure, and you'll cause evolution to occur. So reservoirs need to be kept as low and low nutrient as possible to reduce their effects. I mean, hydropower is meant to be a clean energy, but if you're emitting a lot of methane to generate it, then it's not really, that's not so much the case.
Lars says, hi, thanks for the great talk. Is the low-cost automatic methane sensor accurate enough, or are there any problems with saturation? Did you encounter that, or did the system work without problems? Did you test for that in the lab before using it in the field? Thanks.
Hi, Lars. Nice to hear from you. Nice to see you. To say our system worked without problems would be the biggest exaggeration ever. Our system has been nothing but problems, but we got it working quite well this last summer. It's the end of a 3 to 4 year journey, I would say. In terms of being accurate enough, it's quite hard to believe, given that they are sold for alarm systems in cookers in Japan, that they are accurate enough. And I know that Bestviken and colleagues are preparing another paper that's showing they're even more accurate than they first thought. But you need to do a 3D calibration. They're also sensitive to humidity, so you need to accurately measure humidity at the same time, and then you correct for the humidity in the calibration. In terms of saturation, I guess you can flush them at different frequencies. We flush our chamber every 4 hours with fresh air, and that seems to be fine. I guess with our system for separating diffusion from ebullition, when there's really a lot of ebullition, we probably are underestimating. We're probably underestimating diffusion then with the system we use. But if we use this system, it means we get the data. Rather than having to go through every single plot by eye, because this generates a lot of data. Yeah,
Sure,
so
yeah.
you need an automatic system to process it.
Yeah, and I mean, you get an idea like what happens basically in the system.
Yeah, and we take the median. We've tried taking like just the diffusion estimates from the first 40 minutes, and then from the whole period, and we don't see any difference. So I think it's okay. Yeah.
Okay.
We will continue to test that and try and perfect it. But I'm very interested in making the processing time as easy as possible, so we can actually get the data and use it.
Okay, cool. Yeah, thanks a lot. Yeah, it was great to see you.
Yeah, you too.
Okay, next question from Abdul Raham. I'm sorry because I know I'm not pronouncing the name correctly, but I don't know how to do it. Thank you for this great presentation. What is your opinion about the carbonization of River Delta how we can manage wetlands or ponds to participate in decarbonization.
Oh, that's a big question.
Yeah.
I'm just trying to measure the the gas emissions properly. Management is the really hard bit. Yeah. I think ponds and wetlands can participate in decarbonization, but they need to be in good condition. It's hard to say whether ponds are net sinks of carbon given their emissions, but they are burying carbon. There's no doubt about that. So it's yeah, we need to work a bit to better define the carbon balances of these systems, because of course you have CO2 CIC coming in to a pond. So it's if you have a high input from the catchment, the system is bound to emit carbon dioxide. But it's whether the processes taking place within the pond are making that emission less, or and that the emission of methane is not too high to be higher than the carbon that's buried.
Thank you. Herman asked, thank you, Thomas, for your presentation. Regarding your new device, what methane concentrations ranges cover?
Well, as I said, you can look at the sensors are made by a company called Figaro, and they were developed to be in as an alarm system in cookers, in gas cookers. So they actually cover an extremely high range. They cover to 10,000 ppm. They have it's a very old technology. It's a metal oxide that burns burns the methane when you heat it and put it the amount of methane in the atmosphere affects the signal that comes back from the sensor. So it's a linear signal apart from this interference by humidity. If you look at the papers by Bastviken, you can see the calibration attempts there. It's definitely long enough to cover ebullition from any quantity system over a short period. I mean, I think I don't know if it would work up to 10,000 ppm or something like that, but we're measuring up to 400-500 ppm in really extreme ebullition events. So it covers those ranges. And we of course check it with other systems. It's not, we calibrate it against a kind of state-of-the-art greenhouse gas monitor.
Matthews asks, thank you for your presentation. Did you see the relation about greenhouse gases and cyanobacterial biomass or the phytoplankton community at all? It was talked before. I'm sorry, I messed up with a different time.
No problem, Matthias. As I said in our experiment, there is a calculation with chlorophyll A. We haven't looked at cyanobacteria specifically. But the better correlation was with the derivatives, the DOM produced by chlorophyll. In terms of that was a relatively short-term 8-week experiment looking at methane ebullition in our mesocosm experiment over 8 weeks. In terms of looking at emissions and chlorophyll, we have a data set to look at that now, but I just haven't had time to analyze it. So we'll be working on that in the coming months.
Ineke says, thank you, Thomas, for this great overview. And Tekin asks, hi, Tom, great presentation as always. One question. Are there any alternative ways of quantifying negative emissions other than water concentration? I think we can really quantify negative fluxes using floating chambers. Thank you.
In terms of, hi, Tekin. In terms of the cheap sensors, you can't quantify negative methane fluxes with those because the sensors are not sensitive enough, and we can't quantify them. It's hard to calibrate them in negative yeah. It's hard for us to get lower than atmospheric concentration in the lab without flooding using nitrogen or something. And then the sensors don't work if you take oxygen too low. And we absolutely cannot calibrate them using oxygen, because then they would go kaboom. So yeah, I wouldn't recommend it for that. But then you would need to use some of these other cavity ring down laser-based methods in a chamber. You can calculate the negative fluxes for the CO2 with these sensors, but that's a different thing.
Olga asks, excellent talk. Thank you so much. Are you familiar with IMES by Demian Maher Research Group Enable Continuous Methane Measurements?
This is the Australian group, I think. Yeah, I saw that paper a few years ago. I think it's the same sensor or something similar. Yeah, there are also infrared sensors coming on the market which say they can go down to atmospheric levels for methane, and that would be better. They're a bit more reliable. But the nice thing, it's a happy accident. That the running the methane, the CO2 sensors are infrared sensors, and if they get too humid, they stop working. But the methane sensor, so if you put them on their own, they stop working after a few days. But if you couple them with the methane sensor, it actually warms an element inside to make it run, and this keeps the humidity out of the measuring chamber in the chamber. So it's really a happy accident that they work together. It was a long way of saying I'm not sure that familiar with IAMS, but I'm interested to hear about it. I saw the paper, but I've not seen so much results from it since.
Okay. Shao asks, in Portugal, we have carried out measurements in different hydropower reservoirs, including the degassing, and the variability of results causes difficulties in obtaining the final balance of greenhouse gas emissions. What is your experience? Is there any possibility of testing your methane continuous measuring equipment in one of our reservoirs? What is your opinion about I8A GRES tool? Thank you.
I don't know. I confess I don't know what IHA GRES tool is.
Maybe
Degassing, yeah. I guess you get very variable measurements in the reservoirs because of the management. Can you expand, Zhao?
So maybe he or he can ask, or maybe not.
You can contact me separately if you want to find out about the floating chambers. I mean, we are making more, and we have people good. Yeah, anyone is welcome to come and make some with us if they have time to come and want to make a system. That we are very open to collaborations, and I actually would love more people to use them, and apply them and see if they work in different places.
Olga asked, results you presented nicely demonstrated that reducing greenhouse gas emissions would be an additional benefit of lake restoration. And that's his comment. Enrico says, thank you for your great presentation. What do you think about the relationship between increasing CO2 in the water and species living in the lake? For example, phytoplankton.
So, let's see. Yeah, I don't totally understand the question, but increasing CO2 in the water can be a function of the inputs in the freshwater pipe. So the geology of the catchment and the land use in the catchment, and alkalinity, and alkalinity generally results in increased species richness in plants, at least. I'm not totally sure about phytoplankton, but I have not looked at all about biodiversity of phytoplankton and different types of phytoplanktons effect on greenhouse gas dynamics. I think it's very interesting, and it would be fun to do if blue-greens have a different effect on dynamics, I suppose, as compared to phytoplankton or other types of oligotrophic species. But I'm sorry if I can't answer that question so well.
Matthew, thank you for the presentation. Shao says that he will contact you soon. And Carolina says that, thank you, Thomas, a great presentation. A question, have salinity of a lake an influence over the methane emission?
This is a great question. Maybe Tekin can answer. He's doing his PhD on it. But saline systems have a buffer in that they have a lot of sulfate
which acts as an electron acceptor, which stops the system becoming anoxic in redox terms. So they so you get not such emission, not such large amounts of methane production in the surface waters or the surface of the sediments, but you can get it deeper in the sediments. And I guess with extreme eutrophication, which is kind of one of the things that Tekin is actually studying, is do you get more methane emissions when you get a combination of salinity and eutrophication, a lot of chlorophyll? Do the emissions become high enough? And what about bubbles? Do you get bubbles from saline systems? So they would not be produced very high in the sediments, but maybe if you get high production lower down, you occasionally get bubble emissions. For example, we have these very big deoxygenation events in the seas around Denmark in the late summer, and I wonder if that's causing methane emissions, which is just simply not measured because it's not normally there, but you get extended weeks of zero oxygen. What happens then? It's a nice question.
I don't know if Carolina would like to say something else. Not. Okay, well, thank you very much, Thomas, for this great presentation, for sharing your time with us. And thank you all for attending to this seminar. And we really hope to see you in Brazil in next May. So goodbye and thanks.
Thanks very much for organizing it. It's been a
Thanks a
pleasure.
lot.
Bye-bye.